Okay, so welcome everyone to the last Who's Who of Enzymes of this semester. The topic for today are ammonia lyases. So what are ammonia lyases and what do they do? So if we take a look at their enzyme commission number, we would, well, obviously also from their name, we would get that they are lyases, which means that they are performing a non-hydrolytic removal or addition of a group to the appropriate substrate. Next, we could tell that they are performing a CN bond cleavage or formation. And finally, we can see that the sort of N counterpart is ammonia. So all in all, we can conclude that what ammonia lyases do is they either eliminate or add ammonia to substrates. So if we take a look at this enzyme class as a whole, there are about 7 subclasses that they could be classified into. And the interesting thing is they are, so if we take the criterion that basically forms the definition of ammonia lyase, it puts together this very diverse set of different, mechanistically very different enzyme classes. And so yeah, I will spend some time elaborating on those differences. And as we shall see, they're not only extremely diverse from one another, but when we take a look at each specific mechanism, they're also quite fascinating. In the end, I will mostly focus on aromatic amino acid ammonia lyases, which are probably the most studied ones and are almost synonymous with the entire enzyme class itself. Okay, so starting with aspartate and methyl aspartate ammonia lyases, we can see from the image here, the figure on the left, what they do is they, well, eliminate the amino group from aspartate or methyl aspartate, yield fumaric acid or methyl fumaric acid. So the basic mechanism of how that happens is outlined here below. So there has to be a amino acid residue that acts as a base, which would subsequently form the carboxylic acid analate. And if you take a second to notice, yes, this is like a proper carboxylic acid analate, which we know is quite, yeah, it's not so simple to obtain in chemical conditions, so to speak. We would probably have to use excess of a strong base like LDA or something. So, and then we have a subsequent beta elimination, as we can see here, of the amino group, and the leaving group is also stabilized by hydrogen bonding. And so in the end, we would get a double bond and sort of extended conjugation over this entire system. So how it works more concretely is shown here on the right. So on the bottom right, is the intermediate formed in the active site of aspartate ammonia lyases. And as we can see, the enzyme is, the substrate is kept in place in the active site exclusively by hydrogen bonding, basically, which is quite fascinating. And we see here that this carboxylic acid analate is stabilized only by hydrogen bonding, which is quite fascinating. And in here, serine acts as the catalytic base. And here on the top, we see the active site of methyl aspartates. And if you take a look here, you can notice there's a magnesium ion that instead now stabilizes this analate species, and lysine acts as the catalytic base. So from literature reports, aspartate ammonia lyases are not very promiscuous in terms of substrate specificity, but on the other hand, methyl aspartate ammonia lyases are known to be a lot easier to work with in that respect, and there are some synthetic applications, but yeah, for the sake of keeping it short, I would skip that part, but I would highly recommend checking out this here review that I cited on that topic. Now, moving on to the next interesting class are amino-acyl coenzyme ammonia lyases. Now, if we take a look at what they do here and what the substrates are and the corresponding product, we can see some similarities. So again, we have an elimination of an amine to get the double bond, okay? But the obvious difference is that we have a thioester, so acyl coenzyme A here. And if we take a look at the mechanism, it's actually fairly similar to the last class. So yeah, again, we have a catalytic base, which in here is tyrosine. And basically, again, we get the same beta elimination mechanism, okay? So from enolate, which is again stabilized by hydrogen bonding, we would get beta elimination, and then yeah, the ammonia is again here stabilized by surrounding hydrogen bonds. As far as the enzymes themselves are concerned, so they are fairly rare, and so this beta-alanine coenzyme A, ammonia lyases, is, if I remember correctly, the organism from which it was recovered, so this Clostridium propionicum, which was, I think, found in like mud of San Francisco Bay or something like that. It's the only organism that performs catabolism of beta-alanine, which we see in here, to subsequently make acrylic acid and propionic acid, and hence the name Clostridium propionicum. Okay, so right, moving on to hydroxy amino acids, now called dehydratase deaminases. Now, why are they just simply not called ammonia lyases? Well, because it's a bit disputed if they are completely, I would say, mechanistically ammonia lyases. So if we take a look at the active site, now we have a PLP dependent enzyme. So completely different from what we had previously. And what happens is we see here, so we get first the covalent bonding of the amino acid to PLP, which then activates this alpha carbon to be deprotonated. We get elimination of water. So in this step, it appears that the enzyme is actually a CO lyase, because then in the corresponding step, we get this anamine, which is simply because it's in the tautomeric. Its tautomer is, of course, the imine, which is then non-enzymatically hydrolyzed. So the CN bond lysis is actually not enzymatic technically, but CO bond lysis is enzymatic. But what happens is if you take a look at the overall reaction, you get elimination of water, and then you need 1 molecule of water to hydrolyze the imine, right? So like a net reaction would like not be showing water. So in a way, yeah, you could say that it is a CN lyase. And so far from what I saw, it doesn't really have many synthetic applications. So you would get a carbonyl group at the place where the amino group was. So in this case, a pyruvate. Moving on to the next class, which is again now completely something different, mechanistically again. So yeah, ethanol amine ammonia lyases. So they eliminate the amino group and form aldehydes. Now, as we can see here in the in their active site, they have an adenosyl cobalamine cofactor. So this cofactor is often present in enzymes that mediate radical reactions. And this is also the case in here. So in here we have an outline of the proposed mechanism. So what happens is, so we have a cobalt 3 in in the cofactor, we have a homolytic cleavage of the C cobalt bond, okay? And then the adenosyl radical, which is after substrate binding, is positioned in such a way that it can abstract the hydrogen atom from the from this here alpha carbon, okay? So what happens, what is suggested happens then is this very interesting amine migration, which we basically have this kind of, tri-membered ring transition state, which is which is hypothesized to form. And so yeah, in the end, after the radical is quenched again, we just have elimination of the amino group and formation of the aldehyde like this. So again, completely different from the previous classes, but just taking a look at the final outcome of the reaction, we would classify the enzyme as an ammonia lyase as well. Another interesting subclass, and again, unrelated to previous one mechanistically, are cyclodiamonases. So what happens here is, if we take a look, we have, let's say, ornithine or lysine, depending on the enzyme. And so what happens initially, so it's an NAD dependent enzyme this time, and what happens is what we're used to, some oxidoreductases, C and single bond oxidation. And it looks, if we take a look at the individual steps, it really looks as if this enzyme is more suited to be classified as an oxidoreductase, but the net reaction is redox neutral because we get oxidation and then again reduction, and we have recovery of the NAD, and also within the process loss of ammonia. So again, that is why it is classified as an ammonia lyase as well. So yeah, it's the only ammonia lyase that is NAD dependent, and it actually has had some, I mean, it's a pretty attractive transformation, a pretty attractive way to form, say, piperacolic acid. So it has been applied also on a fairly large scale synthetically. As we can see, there are reports of it of a space-time yield of 0.36 grams per liter per hour, which is not so bad for the lysin cyclodiamonase, and also for ornithine cyclodiamonase. There have been some similar studies. Okay, so finally I reach aromatic amino acid ammonia lyases, which really do deserve a bit of a closer inspection. They're quite interesting. They are definitely most applied in terms of organic synthesis, and they're also quite important. So they are basically key players in many metabolic processes. So for example, in plants, they are one of the key enzymes in the Shikimate pathway. So in plant secondary metabolism, so this pathway is responsible for the formation of many polyphenols, such as flavonoids, coumarins, you see lignines, and so on. So the enzyme that is responsible, so an ammonia lyase that takes part in this pathway is phenylalanine ammonia lyase, which we see does elimination of ammonia to form cinnamic acid. Okay, and then here in the Shikimate pathway, the cinnamic acid is converted into coumaroyl coenzyme A, which is then a precursor to all of these interesting secondary metabolites. So, and then, for example, we have also the histidine ammonia lyase here, which does the same reaction on histidine, which is, yeah, an enzyme that is involved in the histidine catabolism pathway, which ultimately leads to glutamate, which then of course can take part in like a citric acid cycle and so on. So it's a pretty major, pretty major enzyme in also human metabolism. Okay, so how do they work? Now, as fascinating as what they do is, how they do it to me is even more fascinating. And so what these enzymes have in their active site is this methylidine imidazolone moiety. So it is not a cofactor, although it is often called a cofactor, because it is actually formed from the amino acid residues of the main protein chain. Okay, so what happens is, as it's shown here, a sort of cyclization happens to form such quite exotic looking moiety. And so why though? I mean, what is it used for? How does it facilitate the reaction? So there are two proposed pathways for how this happens. So one of them proposes that what happens is this sort of Azamycl reaction, Azamycl-like pathway, whereby the amine of the phenylalanine would attack the beta carbon here. The intermediate would be this imidazole. So we have a sort of thermodynamic driving force, which looks quite clever for this intermediate, which is aromatization here. Okay, and this then further activates this amino group as a leaving group, makes this hydrogen here even more acidic, and it is deprotonated in order to eliminate the amino group. So yeah, now if we really take a closer look at it, there are several ways in which this could happen mechanistically. Of course, it could happen by an E2 elimination mechanism, right? So directly a concerted mechanism, basically, where the subtraction elimination happens simultaneously. It could happen that first an anion is formed, and then that would subsequently eliminate the amino group, or even a cation would be formed in the alpha position here, and then the hydrogen would eliminate. So out of these three options, the last one, so the formation of the cation, is excluded by current research and by experimentation. On the one hand, I mean, it's not very stable, obviously, because it's neighbored to the carbonyl carbon, right? But it also has to do with the, let's say, dipole stabilization in the active site itself. It's not considered likely. On the other hand, between these two, E2 and the E1, there is still some dispute, and it's not quite sure, let's say. So that would be the sort of Azamycl pathway leading to cinnamic acid. Another pathway which was proposed was a sort of Friedel-Crafts mechanism, where the aromatic ring would be attacking the beta carbon here, and then again that would make the hydrogen here to be abstracted more acidic as well. And so yeah, basically just as in an electrophilic substitution on aromatic rings, we would further then have also the re-aromatization and the elimination of the amino group and so on. Now, between these two pathways, the amine pathways is generally considered to be correct. And in some cases, probably the Friedel-Crafts one also happens, but that also depends on the substrate. So it is more likely that it happens if we have tyrosine and histidine, because they are more nucleophilic. It is less likely that it happens with phenylalanine. experiments that have proven that this indeed happens are, for example, on substrates where we would have, say, here in the meta position, a methoxy group. It was shown that the reaction proceeds faster than without it. So yeah, that means probably that there is some electron donate, because of the electron donation into the ring, there is some rate enhancement, and that suggests that this pathway probably happens in those cases as well. But then on the other hand, with substrates such as containing the pentafluoro benzene in here, the reactions proceed as well, and of course there is no way of the electrophilic aromatic substitution happening there. So yeah, we definitely know that the Friedel-Crafts mechanism is not like the exclusive one that happens, but in some cases it might happen to some extent. But in general, the amine pathway is accepted as the one. Okay. So yeah, regarding their synthetic applications, as early as the 1970s, it was already considered for application in industry, and this here shows a transformation that was patented by Pfizer, where they actually mobilized this phenylalanine ammonia lyase and were able to perform the amination of cinnamic acid in quite high concentrations of the substrate. But obviously the real, so yeah, I mean, obviously the enzyme does not only perform the deaminations, of course the reaction is reversible. Usually very high concentrations of ammonia are needed, so like, I don't know, 5 molar, 6 molar, something like that. But the reactions in generally work quite well. And yeah, so the greatest benefit, of course, would not be to make phenylalanine from cinnamic acid, but to make substituted aryl alanines, right, which are easily accessible through their corresponding cinnamic acid derivatives, so to say. So yeah, soon enough, some of those reactions were tried out. And so yeah, this is also I believe, a wild type phenylalanine ammonia lyase from a species of fungus. And we see here that various groups are accepted to some extent. What is very interesting is that regardless of the electron donation effects of the group, we see that ortho substituents are always better than meta, and then meta is always better than para. So this is quite interesting, because then probably electronic effects are not to be blamed, and it is hypothesized that actually there's a steric clash with the para substituents, the greatest steric clash between neighboring amino acid residues and the substrate, which is why it doesn't work quite as well. But interestingly, trying to install substitution on the double bond does not lead to any product formation, and up until today that still hasn't been resolved, or well, up until 2018 for sure, what the one of the reviews that I used here claims. So yeah, that's quite an interesting challenge. It would be a useful, it would be quite useful to overcome. And also, apart from that, nothing apart from ammonia was installed to the to the main backbone. So, for example, methylamine is still not, or any other amine is still not a suitable nucleophile for this reaction. Okay, so in the meantime, of course, there have been more developments, and various heterocycles were also tested as as suitable aryl counterparts in here. And we see I think this is also a wild type enzyme from parsley, simply enough. And yeah, so we see fairly high conversions with different substrates. Here's the pentafluoro phenyl moiety that I mentioned previously also. And yeah, different kinds of heterocycles. What is quite interesting, for example, in this here, I believe pyrazine, yeah, we see some conversion, but as soon as the position of the nitrogen is changed in this way, they saw no conversion at all. And what they postulate is shown here is what actually happens is this, again, azo-Michler reaction happens in a way that forms a stable complex that is shown here. And then basically that acts, this substrate acts as a competitive inhibitor of the enzyme. And that's why the reaction doesn't happen. Okay, so yeah. Well, subsequently, also it was shown to be fairly useful for large-scale production. So a couple of years ago, I believe, yeah, the group of Nicholas Turner in cooperation with Johnson Matthew were able to reach a pretty impressive space-time yield for the production of this here product here in the in this frame, and the transformation was mediated by a phenylalanine ammonia lyase from Anabaena variabilis, which is a species of cyanobacteria actually. So these enzymes are also present in cyanobacteria. What was also this particular enzyme, so from Anabaena variabilis, is known to be quite receptive of different substrates, so it's quite promiscuous and well suited for for different substituted aryl moieties. And what was also a significant contribution in here that enabled such high yields as well was the use of ammonium carbamate instead of ammonia. So what they claim is that basically it acts as a slow release of ammonia because it decomposes right into ammonia and CO2, and so thereby it slowly releases ammonia, and per one mole of carbamate, we would get two moles of ammonia. And on the other hand, we have a fairly low ionic strength, which is also conducive to higher activity of the enzyme. So all in all, quite interesting. I mean, this demonstrates really the also the potential industrial applicability of, not only potential, there have been also applications on a, well, industrial scale of phenylalanine ammonia lyases. So to cut it short, so other things that were achieved, so also it's possible to do a D-amino acid synthesis. The enzymes were also immobilized and I think also run in flow. It's possible to do kinetic resolution, so deracemizations, and chemoenzymatic sequences were also performed. So yeah, obviously these cinnamic acid derivatives are quite easily obtained, probably most easily by the Knevnagle-Dubner condensation, also Wittig reactions, and as well as cross-coupling, because of course if we have these halogen substituted aromatic rings, they're obviously very good substrates for further transformation by cross-coupling chemistry. And that would be it from my side. If you have any questions, I'd be happy to answer.